All right, thanks everyone for coming. I'll be talking about contrasting evolutionary histories between birds of upland and floodplain forests in the Amazon. This is work that I did as part of my PhD at Louisiana State University, although I'm now at the University of Michigan as a postdoc. The geographic distribution of genetic variation within a species can tell us a lot about its evolutionary history. For example, in humans, genetic information from contemporary and ancient samples have revealed a lot about human evolutionary history, including migrations across the globe, demographic changes, and adaptation to new environments. The geographic distribution of genetic variation is driven by a diversity of factors, and these can include extrinsic factors such as landscape change, as HC talked about in the previous talk. In Southeast Asia, the exposed land area has changed dramatically since the last glacial maximum on the left to the present. And you can imagine that this has had a major impact potentially on the genetic variation distributed in this region. Excuse me. And not just in one or a few species, but in many species that are present in the region. But much less well-known is do intrinsic factors of species, such as their ecological or natural history traits, also have an effect on geographic patterns of genetic variation and their evolution. So, for example, do these warblers that have different ecologies, do they, as a result, have potentially different dispersal abilities or demographies that might in turn result in different patterns of geographic genetic variation or evolutionary trajectories? So there's been relatively limited prior work on the impact of intrinsic factors on geographic patterns of genetic variation. Most of this has been in marine systems, but there has been some limited work in terrestrial organisms as well. This is from a paper in 2009 by Bernie and Brumfield, in which they examined a bunch of neotropical forest bird species and their divergences across 3 river barriers. And they found that birds that inhabit the understory on the right in each panel here have deeper genetic divergences across these barriers than birds that inhabit the canopy. And what this suggests is that the microhabitat preferences of these species is having a deterministic impact on their evolution or their genetic divergence. So I sought to delve into this issue of intrinsic factors a little deeper, and the system I used for the study was Amazonian forest birds. Amazonian forest birds are a great system for this because the Amazon basin has the highest species richness of birds in the world, and that richness provides plenty of material for comparative study. So although it might have looked like a carpet of green in the previous photo, the Amazon rainforest is actually a very heterogeneous mixture of habitats. For example, along the big rivers of the Amazon, we find wide floodplains, and in the floodplains is floodplain forest. And you can see it has a fairly linear distribution. Here it's highlighted in black. But in the intervening areas, the dark green areas on the map, or on the satellite image, we find upland forest, which is also called terra firma forest, and that occurs in bigger blocks that are separated by the floodplains. So floodplain and upland forest have pretty different structures and floristics. This is a generalization, but floodplain forest is often more edge-like in structure. It has on the left here, it has a more open canopy, oftentimes a denser understory, and fewer tall trees. Whereas upland forest has a taller, closed canopy, a darker, less dense understory. And as you might expect, given these different structures, the avifaunas of these two habitats are actually quite distinct. So you'll even find in some cases that a species that's present in the floodplain forest, such as the squirrel cuckoo, is replaced in the upland forest by a close relative, in this case the black-bellied cuckoo. So the question for my study is, does the habitat preference of a bird species whether it occurs in floodplain forest or in upland forest, predict the geographic patterns of genetic variation in the evolution of that species. And we might predict that upland bird species will have a greater diversity, greater genetic diversity and deeper evolutionary history. And this could be due to various factors, potentially the larger geographic area of upland forest, as you saw in the satellite image, could produce larger population sizes and greater genetic diversity. River barriers might have a greater impact on upland species because they are set back from the river away from the floodplains, whereas floodplain forest species occur right up to the river margins. Also, the upland forest may be more stable on certain temporal timescales. It's not subject to the dramatic water level changes that floodplain forest is. And to test this prediction, we can compare the genetic diversity and evolutionary history of pairs of close relatives in which one occurs in the floodplain forest and the other occurs in the upland forest. So I used this paired study design and selected 20 genera in which we have a species or superspecies that's distributed in the floodplain forest throughout the Amazon and another that's in the upland forest. So next, I plotted 11 random points across the Amazon, and for each species, I selected the nearest available genetic sample to each of those 11 points. Then I conducted sequence capture targeting 2,300 UCEs and about 100 exons, sequenced on an Illumina HiSeq, and then assembled alignments using a pipeline that's available on GitHub. So just to focus on one of the species pairs, we'll look at the dot-backed antbird that's in floodplain forest and the spot-backed, which is in upland forest. This is the distribution of samples we had for each of those species. We recovered over 90% of the target loci for all of our study species, both exons and UCEs. And when we just start to do some summary statistics from these alignments, you can see that already we're starting to see some differences with more variable sites in the upland forest species than in the floodplain forest species in this case. Other population genetic summary statistics like mutation scale effective population size also reveal differences, larger theta in the upland forest species in this case. As a sort of metric of divergence across the landscape, we use the ratio of sequence divergence between versus the two gene copies within individuals. And that ratio is again higher in upland forest, suggesting higher divergence across the landscape in the upland forest species here. Mean gene tree depth was also deeper in the upland forest species in this case. We also did some model-based population structure, population clustering and cluster assignment. This is using the program Structure, in which you can see the different colors here indicate that we have 4 populations in the upland forest species, but only 1 in the floodplain forest species. And we also used some similar programs and found similar results with Bayesian analysis of population structure and also discriminant analysis of principal components. We can plot these across the distribution of our samples just to get an idea for what the distribution of each cluster is. And then we can also use these population clustering results as tips in a demographic model. And in our case, we used a program called GFOX to estimate demographic parameters under these models. We can estimate things like migration rates between populations, effective population sizes, and divergence times. So we have all these summary statistics and population genetic parameter estimates. And the next step is we have these 40 species, 20 pairs. How do we compare across all our species? So we used 2 methods to do this. The first was generalized linear models with mixed effects. And this we used to test for an effect of habitat association on each genetic parameter estimate while we controlling for which pair of species is in using a random effect. We also did PGLS, or phylogenetic generalized least squares, in that case we used phylogenetic relatedness to control for similarity across species. And I'll show a few of these box plots that just show differences between the 20 species in the upland forest and the 20 species in the floodplain forest on the left. And the dotted lines connect our pairs of species that are joined between the two habitats. So this is just the raw number of variable sites is higher in the upland forest than the floodplain forest, so higher genetic diversity potentially in the upland forest. That metric of divergence across the landscape, sequence divergence versus within individuals, is again higher in the upland forest species than in the floodplain forest species. In most of these graphs you can see there are a few pairs that show a contradictory pattern, but by and large, and these are significant differences, what we might have predicted holds true. The average gene tree depth is also deeper in the upland forest species, suggesting they have deeper histories than the floodplain forest species amongst their extant populations. So we found a series of summary stats or estimates that were significantly different between the two habitats using the GLMs. And then with PGLS, we additionally found that the number of structure populations in DAPC, discriminant analysis of principal component clusters, was higher in the upland forest species than in the floodplain forest species. And so, based on these results, we can say that the birds of upland forest have more genetic structure and deeper divergences in evolutionary histories generally than those in floodplain forest. And there are various implications from this result. We might think that different conservation strategies may be necessary in these different habitats, given the fact that there's greater diversity in upland forest and there's more structuring. They might require different reserve designs or different conservation efforts than the floodplain forests. Also, it would be kind of interesting to think that maybe these differences we see within species might also scale up to differences in speciation over longer evolutionary timescales that might result in disparities in species or trait diversity between these habitats. So I'd like to acknowledge my co-authors and collaborators, Rob Brumfield, my PhD advisor from LSU, and also Alex Aleixo from the Geldi Museum, and Camila Ribas from INPA in Brazil, and also the museums that I got samples from, National Science Foundation for funding, undergraduate assistants, the Brumfield and Roboski Labs for discussion, and many others. Thank you. Yes, Scott.
Do you have any sense in your phylogenetic analyses of, I mean, how much phylogenetic signal is there in, like, the number, the K value in the structure analysis?
There's quite a bit. So yeah, you do tend to see that species in certain families, I mean, I have birds from across the Avon tree of life, basically. So certain families tend to have similar population genetic summary statistics, which is interesting. But we still see this pattern. Yeah?
Do you have a sense of the divergent times between your pairs and whether those habitats were actually available at the time?
Right, so that's this historical perspective is sort of the next round of analyses that I'd like to do. So I haven't looked at that explicitly. I don't have a strong sense for what the divergence times. Most of them are not actually sister species. They're just almost no sister species in which one's in the floodplain and one's in the upland. So, but I should look at that, and I should look at some of these historical possibilities that might also explain some of these patterns.